Are you tired of unwanted eight-legged house guests? Do you find yourself quivering in fear at the thought of dealing with these nasty pests? You may have heard you can deter spiders by placing chestnut seeds around your home, and yes, this does really work. Unless you have a common nut allergy, or you simply don't want to be tripping on nuts all over your house. Well, never fear. We have successfully isolated and purified the compound from chestnuts that eradicates spiders, now commercially produced for use in your own home. Imagine having the spider-repelling power of 1,000 chestnuts condensed into one attractive candle. Now you can set the mood of your home while maintaining a peace of mind. Scientific findings have shown 4 glycosides found in the chestnut endosperm to have toxic effects on insects and microbes. These 4 glycosides are also known as saponins, sticky, soap-like carbohydrates produced as a defense mechanism by a variety of plant species. These saponins are the best candidates as effective spider repellents. First, we need to design a method to effectively isolate our chestnut saponins from a mixture. We adapted a reverse phase HPLC extraction method as originally outlined in the Advanced Pharmacy Bulletin by Abadaya et al. In 2015. We purchased the 4 saponins of interest pre-purified from the National Institute for the Control of Pharmaceutical and Biological Products of China. These were used to calibrate our HPLC conditions. Following calibration, we attempted to isolate the 4 saponin isomers from authentic chestnut seeds. Harvested horse chestnuts were ground and sifted into a fine powder, and the glycoside mixture was purified using an ultrasonic extraction method. This mixture was further filtered through a 200 nanometer membrane prior to loading into HPLC. We purchased a Zorbax 3 micrometer column from Agilent Technologies. We successfully isolated the 4 chestnut saponin isomers, Escin 1A and B, and isoescin 1A and B, with Escin 1A being the most abundant. Confirmation of correct product isolation was performed using thin layer chromatography. This HPLC column purification method will be the basis for later large-scale isolation following recombinant production.
We tested the effectiveness of our 4 purified saponins as a spider deterrent. Using spiders commonly found in North American houses, we constructed an experiment where spiders crossed a saponin-soaked piece of filter paper to reach a food source. We tested individual saponins and combinations of saponins and found that the spiders were most deterred by the saponin Escin 1A. Here we see the desired pathway to our final product, the saponin Escin 1A. Squalene epoxidase and beta-amirin cyclase are known enzymes converting squalene to beta-amirin. The final enzymes to convert beta-amirin to Escin 1A are unknown. We engineered this pathway with known enzymes and candidate magic enzymes, and inserted plasmids containing these genes into a host E. coli strain. Screening steps allowed us to select for the desired magic enzymes, producing our final product. We selected E. coli as a host for this experiment because of several factors, including the commercial availability of competent E. coli strains, the ease of genome manipulation, the ability to create synthetic operons, and the fast replication time of this organism. We also required cells that were saponin resistant, beta-amirin sensitive, and not resistant to the selection marker antibiotics to ensure our screening steps were effective and accurate. We initially considered yeast, as it is naturally saponin resistant, but we would not have been able to create operons, which was a desired step in this experiment. We purchased New England Biolab's T7 Express Competent E. coli with compatible PUC19 vector and then modified this strain to meet our desired characteristics. Saponins have soap-like characteristics and are effective at disrupting the lipid bilayer membrane. The saponin Escin 1A will be produced and will build up inside our E. coli cell. In order to make our E. coli strain saponin resistant, we performed random mutagenesis of the lipid bilayer proteins as a potential way to preserve membrane integrity in the presence of saponins. The technique that we used was error-prone PCR. Error-prone PCR involves uneven concentrations of each of the DNTPs, an incorrect magnesium concentration, and an error-prone polymerase to create products with mutations. These mutant PCR products were then used with the PUC19 vector to make a clone bank. Using robotic screening, clones were exposed to saponin-containing media, and clones that could grow were selected for further use. The enzymes to convert beta-amirin synthase to our end product Saponin Escin are unknown. In order to screen for the correct unknown enzymes, we included a screening step with beta-amirin. If the correct unknown enzymes were present, Escin would be produced. If we had non-functional enzymes, then beta-amirin would build up in the cell. By making our cells sensitive to beta-amirin, this buildup killed the cells, indicating that we did not have the correct enzymes to take beta-amirin to our final product. We chose squalene as the starting point for our engineered enzymatic pathway because it maximizes the amount of intermediate product that follows through to the final pathway. If we started the pathway further upstream, such as in the TCA cycle, we could risk losing a significant amount of intermediate to final products other than our desired product. Squalene is commercially available for purchase, and there are 4 enzymes required to convert it to our final product, Escin. To ensure efficient production of our target saponin, Escin from starting product squalene, we ensured that our enzymes were in close proximity by using enzyme-coupled transport and a protein scaffold. A protein scaffold localized to the inner membrane was used to arrange our enzymes for efficient production of Escin from squalene. Our enzymes, indicated here with letter E, were bound to the scaffold using a linker protein shown in purple. Each scaffold protein has a peptide ligand shown in black, and the linker region joined our enzymes to the peptide ligands. Shown here is a model of enzyme arrangement and our protein scaffold. The scaffold interacts with the local adapter holding it to the plasma membrane. Squalene in the media is taken up into the cell by the reversed MsbA ABC transporter. Squalene epoxidase is coupled to the transporter and converts squalene to 2,3-oxidosqualene. Then, 2,3-oxidosqualene is converted to beta-amirin by beta-amirin synthase located on the scaffold. The remaining 2 magic enzymes on the scaffold convert beta-amirin to our end product Escin. The MsbA ATB binding cassette, or ABC transporter, is a transporter that takes newly synthesized lipopolysaccharides in gram-negative organisms and flips them to the outer membrane of the cell. This transporter is active in E. coli. Since squalene is a long and branched molecule, similar to the components of lipopolysaccharide, this transporter is a suitable candidate for transport of squalene into our cell. We modified this transporter to change the direction from export to import and ensured that the transporter will preferentially import squalene into the cell. Directed evolution was used to modify the MsbA gene. Both the structure and sequence of this transporter are known. We chose to target the residues on the outer membrane side of the transporter, attempting to increase interaction between the transporter and squalene. We used PCR primers with random point mutations in potential substrate interaction regions on the outer membrane. The PCR products with mutations were inserted into the PUC19 vector and cloned into our NEB T7 express competent cells that are saponin resistant and beta-amirin sensitive, creating a clone bank. The clone bank cells were incubated in media without squalene and then subcultured, ensuring at least 2 copies of each clone. Cells from the first culture of all of the clones were lysed, and the lysate was assayed for squalene content with a colorimetric assay, using sulfuric acid and formaldehyde to detect squalene. The procedure was repeated with the second cultures, which had been incubated in media with squalene. The cells were first washed to ensure removal of excess squalene on the outside of the cells and in the media. There was a significant increase in amount of squalene in the second culture, indicating effective uptake of squalene into the cell.
Directed evolution was used to enhance our squalene epoxidase and beta-amirin synthase to maximize their efficiency in converting incoming squalene to beta-amirin. To do this, we used staggered extension shuffling of homologous enzymes. 4 homologous enzymes with similar sequences to our known horse chestnut genes were found through MsbA. For each enzyme, the DNA from all 5 homologs was digested randomly, then PCR'd with very short extension times, giving the denatured DNA a chance to re-anneal to a different homolog. During the PCR process, each elongated DNA strand contained successive portions from several different homologs in random order. Full-length PCR products were screened robotically to find active enzymes. The most efficient ones were then subjected to a repeat of the synthetic shuffling process until the most efficient homologs with the greatest affinity for squalene and 2,3-oxidosqualene were achieved.
We decided to create a plasmid containing an operon of genes responsible for converting squalene into our base saponin, beta-admirin. All these genes are inserted into a plasmid as a functional operon by utilizing sequence and ligation independent cloning. Our operon will consist of a reverse ABC transporter and 3 genes, a squalene epoxidase, a scaffold, and a beta-admirin synthase. The reverse ABC transporter will help transport the squalene into the E. coli cell. The squalene epoxidase will be oxidizing squalene to 2,3-oxidosqualene. This is an intermediate molecule in the saponin pathway. Beta-admirin synthase 1 with the linker peptide will use 2,3-oxidosqualene to produce beta-admirin. This beta-admirin is another intermediate in the saponin pathway. The linker peptide is responsible for covalently linking beta-admirin synthase to the scaffold, and the scaffold will help localize our operon within close proximity of the membrane. We asked Novogen to create a slightly modified PET17B plasmid. The reasons for using PET17B plasmid as our model plasmid is that it produces a low copy number of plasmids per cell. We are interested in a low copy number plasmid because we do not want our cell being lysed prematurely from a high concentration level of beta-admirin that could prove to be toxic for our host cell. One of the most important features of this plasmid is that it comes with a T7 promoter region. This T7 promoter region allows us to express only our genes of interest that are located downstream of the T7 promoter. We do not have to worry about the expression of other genes in the host because the T7 polymerase, which activates the T7 promoter, is specific for only this promoter and will not activate any other promoter in our host cell. This provides us with the ability to control expression of our genes of interest to a very precise level.
The plasmid we utilized was a PET17B derivative with minimal modifications. These modifications included 2 5' complementary regions on the 5' end of each strand on the double-stranded plasmid. The first being a 5' sticky segment complementary to the 5' leading strand of the ABC gene, and the second being a sticky segment complementary to the 5' lagging strand of the beta-admirin synthase gene. We utilized Inverse PCR to prep our plasmid for operon insertion. This required us to develop 2 primers, each going in opposite directions on the same strand, and these primers were found using integrated DNA technologies. And as we can see here, it is this segment that's going to be amplified. Our genes had to be ordered so that complementary regions were flanking both ends of the gene. So for example, the ABC transporter was ordered so that the 5' end of the leading strand was complementary to a 5' region in our vector, and so the 5' end of the lagging strand is complementary to a 5' region of the SQE leading strand. These complementary regions have no thymine residues occurring for at least the first 40 residues from the 3' end. So inverse PCR and SLIC are done first in one reaction. Generation of complementary 5' sticky ends occurs here as well as annealing. The product is then transformed into E. coli, screened, and then sequenced. The T4 chewback reaction generates 5' sticky ends and allows our genes to anneal together and with the vector. Rec A can be added to dramatically increase the recombination event. T4 polymerase will shorten the 3' ends of each gene as well as the plasmid until the first T residue to be integrated is reached. Hydrogen bonding is sufficient to hold the element together throughout the transformation. This is the element that we hope is transformed into E. coli.
Next, we will be taking our plasmid containing our genes of interest and transforming them onto a DH5 alpha E. coli strain. This E. coli strain is an intermediate strain that is only being used here for the screening of successful transformants. We chose this strain of E. coli for transformation because the host range of our PET17B derived plasmid and the host range of another plasmid, which we will be using later on, both allow for the replication of their plasmids within this E. coli. This slide shows the transformation of our plasmids into an E. coli strain. To insert our plasmids into E. coli, we used electroporation. So a quick summary of electroporation is that we start by mixing our DH5 alpha E. coli cells with our plasmid right over here. Next, we're going to run a pulse through our cells at a high voltage to create pores in the membrane of our E. coli cells. Pulsing allows for the uptake of our plasmid by the cell through the pores on the membrane. When you remove the voltage, the pores in the membrane begin to close, and this will trap plasmids inside the E. coli cell. We are then going to take our mixture of E. coli cells and spread plate them onto an ampicillin supplemented media. We're then going to let these plates incubate, and after incubation, we're going to isolate and screen for recombinant colonies. So when examining the agar plate, there are three different possible outcomes. First, a transformed colony containing a recombinant gene. This colony should appear as a white colony because the plasmid is able to insert itself within the LAC-Z gene. A second type of colony that may arise is a transformed colony, which does not have our genes of interest. These colonies will appear as blue colonies because our genes are unable to successfully incorporate themselves within the LAC-Z gene. Lastly, the third type of colony is a non-transformed colony. These colonies will also appear as white colonies like our transformed colony containing our recombinant gene, but unlike them, these will be false positives. In order to screen out these false positive colonies, we performed a colony PCR and sent them in for sequencing. So this is an image of what our finalized plasmid should look like. It will have a PBR origin that allows for the low copy number. It will also carry an ampicillin resistant marker along with our operon encoded between the two ATTL sites. And we are going to be using a T7 promoter to control for the expression of our recombinant genes downstream of the T7 promoter. We can upregulate the T7 promoter expression by using an inducer to increase the T7 polymerase levels. This allows for a precise control over the expression of only our recombinant genes.
Now that we have established the first part of our enzymatic pathway up to the production of beta-amirin, we need to find the final magic enzymes that convert that beta-amirin to our saponin, Escin 1A. We used a homology-based PCR technique, similar to the method originally used by Kishiro et al. To find beta-amirin synthase cDNA. We knew that the enzymes converting beta-amirin to Escin 1A must have acetylase and glycosyltransferase activity. We began by finding several homologous acetylases and glycosyltransferases from other chestnut and walnut genomes on the MsbA database, and then identifying highly conserved regions among them. We then used degenerate oligonucleotide primers based on the conserved regions to amplify cDNA created using RNA from horse chestnuts. This technique produced 17 acetylase genes and 8 glycosyltransferase genes that might be part of our magic enzymes. Once core regions were amplified, we performed rapid amplification of cDNA ends, or RACE, a form of anchored PCR. Specific amplification of each clone using 3' RACE and 5' RACE provided complete DNA sequences.
Candidate genes for magic enzyme 1 and 2 were sequenced using Sanger sequencing. PCR primers were designed from the end sequences of the genes that included restriction cut site overhangs corresponding to restriction sites of the multiple cloning site in the vector these genes were to be inserted into for testing. The genes were then PCR amplified to include the restriction sites. The candidate genes were inserted into PUC19 plasmids that had ampicillin resistance and a LAC-Z multiple cloning site. Different combinations of the two genes were created to find the two correct enzymes that produced our saponin of interest.
The gene combination plasmids were cloned into our engineered E. coli strain. Blue-white screening was used to select for cells with gene inserts. White colonies were selected for the next screening step. The addition of squalene would induce the pathway to create beta-amirin in our cells. Since our E. coli were engineered to be susceptible to beta-amirin, a buildup of this would lead to cell death. However, if the correct group of magic enzymes had been introduced to the E. coli, the beta-amirin could be converted to the saponin of interest, which the E. coli were resistant to, and the cell would survive. The products were extracted from the colonies able to survive in the presence of squalene. HPLC and thin-layer chromatography were used to confirm that the correct saponin was produced. The successful magic enzyme genes were then made into an operon. Custom primers were used to PCR amplify the genes to include a T7 promoter with a restriction site for vector insertion on one end of magic enzyme 1 and 30 base pair scaffold linker peptide sequence overlap on the other ends of the genes. 2 rounds of overlap PCR were used to connect the genes into the final operon that included a T7 promoter, magic enzymes 1 and 2, and scaffold linker peptides to direct the magic enzymes to our scaffold. A pH-SG298 vector with kanamycin resistance was used as the final destination vector that would include both the beta-amirin and magic enzyme operons. A SAC-B gene flanked by ATR1 and ATR2 sites was introduced at the site where the beta-amirin operon would later be inserted. SAC-B gene expression becomes lethal to a cell in the presence of sucrose. This is used later as a counter-selection marker for successful gene insertion. The magic enzyme operon and plasmid were cut with restriction enzymes and inserted into the multiple cloning site of the pH-SG298 vector. The plasmids were transformed into the engineered E. coli strain and screened for correct insertion by plating on media with squalene. Colony PCR and Sanger sequencing were used to confirm correct gene insertions and orientation in surviving colonies. Gateway recombination was used to transfer the beta-amirin operon into our final vector. The operon replaced the SAC-B counter-selection gene by recombining at the AT-L and AT-R sites with the addition of recombination enzymes purchased from Invitrogen. The operon insert in the final plasmid was flanked by AT-P and AT-B scars after the recombination. The final plasmids were transformed into our engineered E. coli strain. The E. coli were grown on media containing kanamycin and sucrose to activate the SAC-B lethal gene. Only cells that had the destination vector with the gene insert replacing SAC-B would survive. From the surviving colonies, colony PCR and Sanger sequencing were performed to confirm the genes were correctly inserted into our plasmid.
Now that we've successfully engineered our recombinant enzymatic pathway in E. coli, it is time to prepare for large-scale industrial production. We decided to utilize a fed batch fermenter, as this is the most sophisticated bioreactor approach and allows for the optimization for recombinant production. We developed a stirred tank fed batch bioreactor, coupled to a fully integrated reverse phage chromatography purification column. Following the addition of our anti-spider E. coli, along with squalene supplemented media, the fed batch tank is equipped with sensors to monitor changes in oxygen, pH, and temperature, while measuring the amount of biomass as saponin and culture volumes increase. This sophisticated monitoring system allows for the accurate addition of substrates and buffers to ensure maximum recombinant production. Kanamycin is periodically spiked into the media to ensure the selection of E. coli retaining our final constructed plasmid. Stir paddles ensure an even distribution of media and products throughout the fermentation tank, and can be utilized since our E. coli are small and have flexible membranes. Shear force generated from stir paddles can cause complications when utilizing larger organisms, such as Saccharomyces. As previously mentioned, the use of a fed batch fermenter allows for the optimization for recombinant production. Optimal growth conditions were adapted from an article by SLIC et al., published in Biotech and Bioengineering in 2004, highlighting a similar method for recombinant pyruvate production. Once biomass levels have reached threshold, Cells are mechanically lysed using a French press mechanism equipped with a 500 kilodalton size exclusion mesh. The press compacts cellular debris along with non-soluble metabolites to the bottom and out of the tank, leaving our saponin containing supernatant in the 7.5 liter chamber. The supernatant then passes through a hollow fiber ultrafiltration unit, purifying any protein left in the mixture. The cell-free broth, now cell and protein-free, is further filtered through a 200 nanometer membrane prior to loading into a liquid chromatography column. Similar to the HPLC method described at the beginning of this presentation, we utilize the Zorbax 3 micrometer column purchased from Agilent Technologies. We, however, custom ordered the column for large-scale product isolation. This allowed for much greater sample volume input, with all other conditions remaining the same. Based on the success outlined by SLIC et al., we postulate an approximate saponin yield of 500 grams per liter cell culture, which works out to be about 2.5 kilograms per 7.5 liter batch, grown over about a 15-hour time period. With our successfully isolated saponin, we infused the extract into decorative candles. The candles were then subject to a variety of quality control tests, such as the candle's effectiveness relative to that of pure saponin, candle toxicity, both to humans and pets, along with the effects of long-term storage, as we would like our product to have a marketable shelf life. We conducted some market research and found many different chestnut-scented candles and sprays, including this product shown here. As a possible business option, we considered the possibility of licensing our spider-repelling technology to companies that produce candles and other air-freshening products. This synthetic saponin product will revolutionize the spider repellent technology and fragrance industries and has massive earning potential when marketed effectively. We have a projected product launch date of Christmas 2017, so don't miss out and pre-order your candle now.